We all know that X and Y chromosomes determine sex in mammals, but more specifically, it's the SRY gene on the Y chromosome that makes a developing fetus male. Sometimes, although it is rare, the SRY gene will be crossed over onto an X chromosome in the testes of the father. The fetus with a Y chromosome that is missing the SRY gene will be born atomically female despite having a Y chromosome. But is this how it works for all species? For many reptiles, it is all about hormone production. If testosterone is converted to estrogen, the fetus will develop into a female instead of male. This whole process is dependent upon the temperature and whether the enzyme aromatase is activated or not. Shown here is the structure of human aromatase. Aromatase belongs to the cytochrome P450 family of proteins and catalyzes the synthesis of estrogen from testosterone. This protein expresses in many tissues. However, it is localized mainly in the ovaries. Here is the mechanism of conversion of testosterone to estrogen that is catalyzed by aromatase. It requires NADPH as a cofactor in order to proceed. So, what causes the activation of aromatase? The answer is different incubation temperatures. There are differences in species on what temperatures result in male or female offspring. One way that researchers try to learn more about the process is by introducing estrogen to the developing fetuses in the egg. This is done by coating the shell with small doses of estrogen and seeing what sex develops. They found that even at male temperatures, the egg coated with estrogen are born female. This shows that the presence of estrogen is all that is necessary to form a female regardless of temperature. Therefore, in natural conditions, the action of aromatase is essential in generating female offspring. How can temperature activate an enzyme? We don't know for sure yet, but there are some studies. In one study, researchers have been investigating whether temperature affects methylation of promoter region of aromatase. This study was done on European sea bass in the aromatase promoter region CYP19A. In sea bass, higher incubation temperatures produce males. In the first 60 days of life, they subjected some to high temperatures and others to low temperatures. Females at lower temperatures had the lowest methylation at the promoter region and a higher methylation at higher temperatures. The same applies for males. Methylation can act to prevent transcription, and the results of this study suggest that connection between temperature and sex differentiation is through the change in methylation. Another study done on red-eared slider turtles showed similar results than the European sea bass. Even though we know quite a lot about this topic, there are some unanswered questions, such as how temperature increases methylation and how temperature can activate an enzyme.